The stem cell review is presented by Celgene Cellular Therapeutics.
I've been working with in the area of tissue engineering since 1983. In fact, Jay Vacanti, one of my close collaborators, he's a head of pediatric surgery at Mass General Hospital, and I started talking about ways to create new tissues and organs. And originally, of course, we started with various kinds of cells like skin cells, cartilage cells, bone cells. But as time went on and stem cells became available, that became very attractive. And so starting in about 2000, we took even like human embryonic stem cells, and Romet-Lemonne in our lab, she's now professor at the Technion, worked out the first ways of converting them to endothelial cells with the opportunity to, say, someday create new blood vessels. Bioengineers play an important role in regenerative medicine and stem cell, potential stem cell therapies in a number of ways. First, you need to isolate the cells, which is an engineering challenge. Then you may need to expand them and purify them, which is also an engineering challenge. There's several strategies that people are looking at in terms of taking cells and making a tissue or organ out of them. One is what I'd call immunoisolation. And there what you do is you want to take the cells, it could be a stem cell or it could be a beta cell that you've taken the stem cells and converted it into a beta cell to treat a diabetic. But a lot of times those cells could be rejected by the body. So what you have to do is encapsulate them with a material. And the idea of this material is one that has pores. And the pores, I'll just pick diabetes as an example, the pores should be large enough so that substances the size of insulin and glucose can get through, but they shouldn't be so big. In other words, they should be able to prevent antibodies from getting through. They should be able to prevent immune cells from getting through that might attack and destroy the stem cells or the beta cells. So figuring out the right material, the right porosity, the right design is key for this immunoisolation approach. If you had a scaffolding, what you could do, which could be a material that you put the cells on, that can help guide the tissue. So guide the cell development. For example, if you take cells and inject them into the body at random, they probably will not form a tissue or organ. But interestingly, if you put cells close enough together, they actually have the ability to communicate and create certain tissue structures. A very, very striking example of that was a study they did at Berkeley a while back where they took mammary epithelial cells, put them close together in vitro, and they actually made acini and made milk. So the idea is that if you were able to design a scaffold that the cells could be on, the cells would be close enough together, they would actually form matrix, they would proliferate. Over time, the scaffold itself might dissolve, and then you'd be left with the cells in pure matrix. And that's been a strategy that people have used to create cartilage and bone and skin. And there, in some cases, you might have to worry about rejection if you, but in other cases you may not, if you, depending on the cells you use and how they were protected, like whether you used immunosuppressive drugs or not. But this is an approach that's now being widely used for a variety of tissues and organs, and of course engineers have played a very central role in developing that. Finally, you would want engineers to help in the monitoring of the success of this tissue engineering or regenerative medicine approach. In other words, what types of markers are you going to look at? What type of criteria would you use to judge success? And so the engineers would also be involved in the monitoring and developing the right ways to monitor whether the stem cells were being used properly. If you're trying to do heart tissue, I mean, you can't just inject the cells and hope they'll go where you want. I mean, you have to have some type of design, some type of approach, a catheter or something else that would take them to the heart, and then you'd have to have some type of gel or scaffold that would keep them there, that wouldn't cause damage to the heart, and that would allow the cells to grow and proliferate and do their job. Another area we've done a lot of work on in collaboration with Evan Snyder and Ted Tang, and this was done actually also by Aaron Levick, who was one of my former students, now professor at Case Western, has been to take neuronal stem cells and use them as a possible means for spinal cord repair. What we did in a number of studies is made rats paraplegic, and then we created a polymer scaffold that actually kind of mimicked the gray and white matter of the spinal cord, and took the neuronal stem cells and put them in, and they actually enabled the rats who were paralyzed to pretty much walk again. I mean, now we're actually in primate trials, so it's been very exciting. The FDA will look favorably upon things that are safe, effective, and innovative. And I think a scaffold probably helps on all those things. So I would think that generally it would be looked upon as a positive thing. The regulatory agents would look at this as a combination product. It's a combination of, you know, they might have input from both the device type people as well as biologic type people. So, but it would be viewed as a combination therapy. I think the most important thing for the companies is efficacy and safety. I mean, I think the fact that it's a combination product certainly is not a bad thing, but the key, however you do it, is to make it safe and effective. How you're going to monitor and how you're going to monitor success, how you're going to judge success is key. And so I think, you know, the question is what endpoints are you looking at? And of course that'll vary from tissue to tissue and organ to organ. They're all going to be different. It's important to realize that cell therapy, stem cell therapy, regenerative medicine, I think offer opportunities for clinical treatments and business opportunities that you just can't do with drugs. You can't make a new liver with a drug, and you can't make a new pancreas with a drug. You can't cure somebody who is paralyzed with a drug, and we could go on and on. So I think that cell therapies are really potentially revolutionary. I mean, because they could offer the potential, I mean, we're not there yet, but they offer the potential for treatments that you're just never going to have any other way. And they're some of the biggest diseases and problems out there. So I think it's a huge challenge, but it's also a huge opportunity.
At Celgene Cellular Therapeutics, our proprietary placenta-derived stem cells hold the potential to treat a range of serious diseases. Our unique processes for differentiating and producing treatments from this rich source of stem cells will enable us to deliver these therapies on a scale similar to today's biologic remedies. Proud to be at the forefront of this transformation of modern medicine, we are Celgene Cellular Therapeutics.